Okay. Hello, everyone.
Okay.
My name is Javier Jofre. I'm the Corporate Strategy and Marketing Director of Atens. Today, and during all the sessions that we are doing during the celebration of Fruit Attraction LiveConnect, I will be moderating the webinar. First, I would like to thank everyone who is joining us today in the live stream. We have prepared this session with great enthusiasm in order to continue spreading our work of 25 years of experience as developers of innovative, effective, and biological solutions that provide solutions to the problems that the agro industry is facing every day. During the celebration of Fruit Attraction LiveConnect, we have prepared a series of technical conferences which will cover a wide variety of crops, such as red fruits that we did last week, cereals that we're having today, and next week we will have horticultural crops. Always with the purpose of giving a methodological approach of the use of microorganisms, as well as ending each session by sharing several experiences obtained by both our own trials and also trials carried out by some of our clients. Today we will share a session dedicated exclusively to our seed treatment products based on Mycorrhiza, Trichoderma, and PGPR bacteria. I'm talking about the COVERON line and the Team Extensive products. To develop this topic, we will have the participation of Mycorrhiza, Technical Director of Atens, and Roberto Longo, Technical Commercial Director of Italpolina, Italy. Italpolina is a leading player in the organic fertilizers industry with 50 years of experience, offering a wide range of biotechnological and certified solutions for a more sustainable and modern agriculture. To this day, Italpolina's footprint continues to grow with an international presence that comprises more than 80 countries. Most recently, Italpolina opened a new US manufacturing facility in Anderson, Indiana, which is completely dedicated to the production of natural biostimulants based on 100% vegetal amino acids and peptides. In terms of microorganisms, Atens is the official supplier of the microorganisms-based products of Italpolina. They have a strong distribution network of these products in several markets, more importantly, Europe and the US. Mycorrhiza and Trichoderma products of Italpolina are exclusively manufactured by Atens, and some of them include in their composition exclusive strains developed for them. Gorka will begin the session with a brief review of our company, continuing with a technical review of our Mycorrhiza and Trichoderma, and ending with different trials and success stories in cereals. After this, Roberto will take the lead, explaining his experience using the seed treatment. Please note that the COVERON line used by Roberto is manufactured by Atens and is an homologous of our COVERON line of products. Please feel free to send your questions using the chat available in the LiveConnect platform. We will try to give a response at the end of the session. Without any further delay, please welcome Mycorrhiza.
Thank you, Javier, and welcome everybody. Welcome all the attendees in the Fruit Attraction LiveConnect. My name is Mycorrhiza. I'm Technical Director in Atens, and today we will cover the seed treatment products, the solution from Atens for the seed treatment. Let me share my screen with all of you. And as I told you, we are going to cover the seed treatment solutions. But first, let me tell you about Atens, our company. Atens is a family-owned company founded in 1994. That means that we have more than 25 years of history. Atens was born from the research and development in order to develop a mycorrhiza for sustainable agriculture. Nowadays, we are the world's largest producer of mycorrhiza in vivo and Trichoderma in solid medium in a bioreactor. So, as I told you, Atens was born from the research and development, and it's completely linked to the R&D right now. Please let me show you some of the facilities that we have in our lab. Our lab is called the NGALAB. That stands for Next Generation Agronomics. In that lab, we have machinery like this, like the MySec from Illumina, from a sample of soil, we can sequence massive millions of DNA fragments in a single process. So we can discover with this MySec what are the populations in the soil, what are the relationship between these populations affecting the health of our plants. So we are also the first company to sequence the genome of our Trichoderma strain, the TK7. I will talk to you later about this. And we also have in the NGALAB this LCMS, like a chromatography linked to mass spectrometry. From a complex sample of leaf or fruit extract, we can separate all the components from the liquid chromatography, identify all the components with the mass spectrometry. That means that when we apply our solutions to the soil, we can alter the plant physiology and the soil microorganisms. We can obtain that outcome from higher yield or higher production in our plants. But if we don't understand what happens inside of the plants, and we don't know what are the metabolic pathways altered, we cannot conclude about the mechanisms in which our microorganisms work. So this tool is essential for us to improve our microorganisms and to develop new microorganisms to help plants. And as we have in our laboratory these facilities, we have been able to develop two inventions. The first one is related to the seed treatment, so it's going to be crucial in this presentation. It's very difficult to bond, to coat the seeds with spores of Mycorrhiza and Trichoderma, so we developed an invention to attach that spores of Mycorrhiza and Trichoderma to the surface of the seeds, and that invention was resulting in a Spanish patent application, also an international one, an European one, an American, and a Brazilian one. Also, we have a second invention that is related to reducing water stress in plants. It's related to a plant fiber that we include in our products in tablet format, tablet to apply during transplanting of the plants directly in the hole. Well, that tablet contains the plant fiber that also it's able to retain the water around the rhizosphere and make that water available for the plants and for the microorganisms in the rhizosphere. It also, the plant fiber can also prevent the leaking of the fertilizer that is included in the tablet. Well, with this invention, we have a European patent, an international one, American, and a Chilean one. So, as you may check, we have our passion for research, and the other pillar of Atens is the passion for the quality. We tend to invest the best we can do to improve the quality of our products. That's why in these last 25 years, we have developed a mycorrhiza production system using greenhouses, I show you later, and a solid-state bioreactors for Trichoderma productions. As well as we have imported the cosmetic technology in some machinery just to mix the formulation in our products, or the pharmaceutical technology to make the tablet format products. Also, it's very valuable from the farmers that our products are zero residue with no security periods, and they respect the auxiliary fauna. And we are also signatory of the Pact for a Circular Economy, and we are focused on the food challenge of 2050. That means that in a growing population planet, as in which we are, we try to produce better in a more sustainable way for more population. We also have certification for quality management and environmental management, as well as energy management, and we are fertilizer manufacturers. All our products have organic certification, and part of them are in the ombre list of the USDA. But let's move to our microorganisms, and we can start with the first one of them, which is the mycorrhiza. Mycorrhiza, everybody knows at this point, it's the association, mutualism between a fungi and a plant. The fungi can colonize the roots of the plant and can enhance the absorption of certain nutrients and water in the plant. So it can explore better the soil with a higher volume, but also it can increase the phosphorus intake, as the mycorrhiza has phosphatases that can extract directly from the soil matrix the phosphorus in a basic pH soil. It also improves the water status of the plants, as they can explore more soil, and the mycorrhiza hyphae are 10 times thinner than the regular root. So when during a drought episode in which the smallest pores has the reservoir of water, this water is available for the mycorrhizal plants, whereas it's not available for the naked root plant. It has also been demonstrated that the mycorrhizal plants have higher antioxidant enzymes activity, as superoxide dismutase and ascorbate peroxidase, as well as glutathione reductase. So during the normal metabolisms under radical oxygen species production, the mycorrhizal plants can scavenge those species faster than the non-mycorrhizal plants. And also they have more resistance to abiotic stress, such as low and high temperature and heavy metal presence in the soil, particularly the copper. And they also show a higher resistance against some pathogens. Mycorrhiza, they don't kill nematodes, they are non-nematicides, but during a nematode attack, as plants can have a better water status, they can resist or tolerate better that pathogen attack. Well, in Atens, our products based on mycorrhiza, they belong to one of these three groups in the Spanish legislation, Spanish registry. The mycorrhiza, they are a mixture of microorganisms or fertilizers with microorganisms. And the products based on mycorrhiza in Atens, they are the combination of two different strains. The first one is the Rhizoglomus irregulari BEG 72. It's one of the best known and most studied species. I can tell of that because 10 years ago, when I was working in academia, I was working in a project about alfalfa under climate change conditions. Alfalfa in symbiosis with nitrogen-fixing bacteria. And at that point, we want to add a third partner in this symbiosis. And we made a bibliographic research to know what is the best known mycorrhiza in the scientific literature. And BEG 72 was, by way, the largest with the largest amount of publications. So we include that in this study with very good results. This strain was isolated in collaboration with IRTA, the Catalan Research Agronomic Center. On the other side, we have the Funeliformes mosche, BEG 234. This is a different mycorrhiza with largest spore size, and the studies focus with this mycorrhiza. They talk about how the plants can resist the effects of water stress, drought, and the large amount of phosphorus that Funeliformes mosche BEG 234 can dissolve in the soil. This strain was isolated in collaboration with the ICIA, the Canarian Institute for Agronomic Research. But these two strains have a lot of potential, but we want to produce them in a way that this potential can be maximize. And that's why it's important, the mycorrhiza production system. To produce the mycorrhiza, we have different options. The first one that we can claim, it's the open field conditions. We have a field with the host plants that we can inoculate with mycorrhiza, and at the end of the season, we can take over all the substrate and we can filtrate it and take all the spores of mycorrhiza to prepare the formulations. Well, as all the farmers may know, there are no 2 consecutive years with similar conditions. So this system will lead to a regular production of mycorrhiza, and that's a big problem from the point of view of the industry. And also they has difficult spore collection system, and the worst of all is that we have risk of contamination. If a pathogenic fungi arrives to our open field, it can contaminate the production, and we cannot work with that. So in order to limit the contamination, we can produce the mycorrhiza in vitro. Here we can ensure that there is an absence of contamination in these petri dishes that we can share between the carrot roots, transformed carrot roots in one side, and the mycorrhiza in the other side in an arsenic medium. Well, we can collect the spores from this petri dish, and it's a high spore production, but these spores are morphologically very different from the open field producer spores. So these spores with the less content of DNA inside, they have been proved to be very low, to have very low level of infectivity. And also these spores have low spore life, which is a problem for the industry. So in Atens, we have opted for a third way, which is to produce our mycorrhiza in the greenhouse. Here in the greenhouse, we can have our host plants growing in a substrate controlled with no contaminations, with absence of pathogens, and we can have a high spore production. Spores that are morphologically like the open field spores. And also these spores show a higher spore life than the in vitro produced spores. So let me show you a little bit more about our greenhouse production. This is our greenhouse, our host plants growing in tables, not in the soil. Here we have an annual production of mycorrhiza. The greenhouse is completely automated, so the production of spores is the same each year. All these conditions are part of this high quality search that we want in Atens, and also searching for the standardization, which is, I have to reinforce that, which is essential for the industrial production of mycorrhiza. We have our quality controls during all the production of mycorrhiza, but to make sure with a double check, every batch is also analyzed by the IRTA as independent center. So we have our quality control, we have the IRTA quality control, and we can check if both of them match. But one of the advantages of this in vivo system is that together with the mycorrhiza, there are also some bacteria that have been reproduced. 2 years ago, we published this paper in Frontiers in Microbiology about the community of bacteria surrounded our mycorrhiza. And we discovered 15 strains of bacteria that have properties of PGPR. That means plant growth promoting activity. Why they have this PGPR activity? Well, because some of them are nitrogen fixing bacteria. Some of them can solubilize the phosphorus or produce the indolacetic acid. Also, they can produce siderophores. That means molecules that can be secreted to the soil and then mobilize the iron iron. So they can incorporate the iron to the cytoplasm, and that iron is available not only for the bacteria, but also for the plant. Well, if we move forward and talk about the Trichoderma, Trichoderma is a very different fungus from Mycorrhiza. Trichoderma is a saprophyte. It feeds on organic matter, so it can be established at the root level in the rhizosphere and feed on the plant exudates. One of the main characteristics of Trichoderma is that it competes with other fungi for space and nutrients. Part of the problem from the pathogenic fungi is that they are very nice competitors. So they can establish at the root level, and it's very difficult to make this space or the nutrients in the soil available for other beneficial fungi and bacteria. So if we can add Trichoderma to our soil, we can compete against these pathogenic fungi, and we can reduce the space for them and the nutrients, all the resources in the soil. So we can have half of the battle win. So as I told you, Trichoderma can rapidly colonize the soil and the rhizosphere, and it also promotes the defense of the plants. The induces systemic resistance. The plants in presence of Trichoderma, also with Mycorrhiza, they can have their jasmonic acid metabolism pathway exalted, so they can respond better and in a more efficient way to the real pathogen infection with more efficiency. And Trichoderma also stimulates the root development, like with volatile organic compounds, or even by the synthesis of auxins. It can also synthesize siderophores as well as the bacteria, and it improves the fertility and the vitality of the soil. Here in Atens, we can produce two kinds of strains. The first one is the Trichoderma koningil, TK7, is one of the most known Trichoderma strains. It also has all the biostimulant properties that we have talked about in the slide before, and it promotes the defense of the plant. This strain is registered as a fertilizing product in the group 4 for non-mycorrhizal microorganisms under the Spanish registry. The other strain is the Trichoderma troviridae 8010. This is a strain in the phytosanitary registration process. In fact, it is currently in the last phase of approval as an active substance in the European Union as well as in the USA. It's a very aggressive strain, and it can be used against a wide range of fungal diseases of soil and wood. For example, Fusarium, Sclerotinia, Botrytis, and Esca. Well, as it happened with the Mycorrhiza, with the Trichoderma, it's the same. The system of production, it's essential to guarantee the highest quality. So this is the solid state Trichoderma fermentator that we have in Atens. It's a vertical 3-module one. Here you have the real fermentator. Here you have the cartoon in order to show you how we control the growing Trichoderma. Here we select the Trichoderma koningil TK7 at industrial scale in a completely automated process. This process, from the cleaning to sterilization, fermentation, and drying, it takes more than 40 days to complete. It takes more than 40 days to get the more mature spores of Trichoderma that can ensure that our Trichoderma is going to be mature enough to establish well in the soil. Here in the Trichoderma fermentator, we have no pollution, so we can maintain the quality that comes from our internal quality control and external one. We have a laboratory that certifies that our quality control is okay. So for the seed treatment, we have two options. The first one, it's called the covenant line products. That kind of products are for the industrial treatment of seeds. They need a dedicated machinery, and they are under a powder formulation to dilute in water, and then to apply a liquid to apply them a liquid to the seeds. Well, this covenant line is focused on the seed treatment industry and the big producers, big farmers that have access to this dedicated machinery. On the other side, we have another product which is called Team Extensivo. This is for the treatment of seeds in the field. The main characteristic of this team is that it's easy and is for flexible use, and they have no requirements of dedicated machinery. The formulation of the Team Extensivo is granulated for dry application directly in the hopper. So it's essential to distinguish these two lines of the products. So if we start with the covenant line, I'm going to present you what kind of industry is dedicated for this product. As an example, in the picture, the Consorzio Agrario della Emilia in Italy, it's an association of farmers of reference in whole Europe. They have all these machinery and all these seed treatment facilities that can apply this kind of products. We have different products for the covenant family, let's say, but first, the question is why to use a microbiological coating instead of the chemical coatings that are in the market? Well, they are more natural, they are not pesticides, and they can hence reduce the handling cost. I mean, if you coat the seeds with this kind of microbiological, they are not required to be disposed by seed companies. Everyone can have their own seeds treated and they can dispose in the way that they consider. And also they are multifunctional products. I mean, they have all the properties from the Mycorrhiza and Trichoderma, and this is more important, these properties are going to be carried out during all the life of the plant. They are not going to act during the first stage of development, but they are going to be in the soil for the whole cycle of the plant. So the first one of the products, it's called simply Covenant. It's the objective to incorporate the benefits of such microorganisms, Mycorrhiza and Trichoderma, directly from the showing. And it has a very balanced concentration between the concentration of Trichoderma and the concentration of Mycorrhiza. The dose, it varies between 100 and 200 grams per 100 kilograms of seeds. The other option is Covenant Trichoderma, which is more balanced to the side of the Trichoderma than to the Mycorrhiza. It has Trichoderma with more concentrated than Mycorrhiza. And the dose, it depends on the species. The dose in cereals, it's 20 grams per 100 kilograms of seeds. And the dose in legume species, it's 40 grams for 100 kilograms of seeds. Then we have two different covenants for specific species. And I will show you why. The first one, it's called Covenant SOJA, which means soybean. It's designed precisely for the soybean because it includes Bradyrhizorium japonicum, which is a nitrogen-fixing bacteria specific for soybean. The dose, as in the previous case of Covenant, it varies between 150 to 200 grams per 100 kilograms of seeds. And the other product dedicated to a specific plant is the Covenant ARROZ, rice. Covenant ARROZ, as well as the Covenant Trichoderma, it's more balanced to the side of the Trichoderma than to the Mycorrhiza. And in this case, the dose, it will depend on the application methodology. If the rice is going to be shown in water-logged soil, the dose is 20 grams per 100 kilograms of seeds. Whereas if it's going to be shown in a dry land soil, 20 to 30 grams per 100 kilograms of seeds. Well, let me finish with some examples of tests that we have made using this product. And the first one is the soybean treated with Covenant SOJA. And this was made in Brazil. It makes all the sense to make this kind of test in one of the better producers of soybean in the world. The company was made by AgroTarget in the state of Minas Gerais, and the treatment was applied 2 hours before planting. The dose, the normal one, 150 grams per 100 kilograms of seeds. So from the early development, and these results are from 27 days after plantation, the treated plants show a higher stem and leaf matter, as well as higher leaf area development. This is essential because the leaf area is going to provide all the carbohydrates for the mycorrhiza and for the bacteria for the nitrogen fixing. Well, 64 days after planting, all these results were the same. So the enhancement of the stem matter, the leaf matter, and the leaf area. So as I told you, the benefits from the microbiological coating, they can be shown during all the cycle of the plant. So what happened at the end of the season? Well, we have a yield of 3,896 kilograms per hectare in the control treatment and 4,300 in the Covenant SOJA treated plants. That means more than 10% of the yield increase. This is a test about Covenant ARROZ in rice. It was carried out in Spain by Agricampo. So we have two different cultivars of rice. The first one is type Perla, which was shown in dry conditions, and then waterlogged from day 45. And the other one, the other cultivar is GLAUDIO, which was shown in waterlogged land from the beginning. So type Perla was applied to a dose of 30 grams per 100 kilograms of seeds, and GLAUDIO was only 20 grams per 100 kilograms of seeds. I haven't told you, but all the Covenant line products comes with a green stain, so you can check whenever your seed has been treated with the microbiological coating, and you can check that this coating has been uniform for all the seeds. Here are some pictures of the dry showing of type Perla. And this is type Perla in the first stages. So you can check that the Covenant treated plants are bigger and they have a better development than the control ones. So that's one of the keys. The early development, it's a better development with the microbiological coating. This is the end of the season, the harvest time, and we have some results about the yield. So for type Perla, The control area, 3.7 hectares, and the dry rice production was 30,000. It remains a yield of 8,300 kilograms per hectare, whereas the Atens treatment, the area was 3 hectares, 26,000 of kilograms of dye production, and the yield was 8,700 kilograms per hectare. That means that 456 kilograms more with the Atens treatment. And translated to euros, it means 135 euros more of benefits per hectare. And as you can see, similar results were obtained in the other cultivar, CLAUDIO, with a control yield of 8,300 kilograms per hectare against the Atens treatment with 8,500 kilograms per hectare. That means 416 kilograms more with the Atens product and 128 euros more of benefits per hectare. So now we're talking about Team Extensivo, the other option. As you remember, the treatment of seeds in the field, AC and flexible and granulated. This is the product, more thing for every farmer. There is no need of dedicated machinery, and well, probably the best option, because you can coat your seeds, your grain seeds directly in the hopper. So, as in the case of COVERON, it has a very well balanced concentration of Mycorrhiza and Trichoderma, and the dose for wheat, barley, oats, corn, sorghum, every seed, it varies from 0.5 to 1 kilograms per hectare. And let me show you a couple of trials that we have carried out. This is in the Centro Sperimentale ASSAM in Italy, that we confront the Team Extensivo with a chemical coating. And at the end of the season, I haven't told, it's a wheat cultivar CLAUDIO, it's a durum wheat. We have a difference in yield, 4.66 versus 5.71. That means an increase of 22.5%. Of course, there is an increase in the spike number, but only about the 6%. That means that our coating not only has produced more spikes, 6% more, but that our spikes and our grains are more filled and are bigger with the Team Extensivo. This is the last trial I'm going to show you. It's Team Extensivo in barley. That test was carried out in Spain, and you can see the difference of size in spikes in barley, the Team Extensivo treated plants versus the non-treated plants. To show you some numbers, the control plants, they reach to 2.38 tons per hectare, and the treated plants reach 3.14 tons per hectare. I'm going to show you in a table 58.14 of total production in an area of 16.02. That means a yield of 2.58, and Team Extensivo 28 in an area of 9. That means 3.14. That means, if you calculate that with Team Extensivo, there is a 52% increase in yield in those plants. So I have finished my part, and I am very proud to give the word to Dr. Roberto Longo. Dr. Longo is is the technical and commercial director in Talpolina, which is an historical customer of Atens. They produce, we produce microorganisms for them, and they have their own registering microorganisms. So let me, it's an honor to be here with Roberto Longo. So I'm very interested in what he's going to tell us.
Okay. Thank you, Gorka, and good afternoon at all. It's a pleasure for me to present our large experience on seed dressing seeds on wheat, rice, and corn and soybean. Wait a moment, I show you my presentation. Wait a moment. Okay, well, you see? Okay. Seed dressing experience on wheat, rice, corn, and soybean with the COVERON Trichoderma. Iterpolina has been using COVERON in these last 12 years on different crop, wheat, rice, corn, soybean, and we are starting to use on horticultural crops. There is a big attention on these products because it meets the needs of modern farmer. I start to explain our experience on wheat. The needs of Italian farmer are this: use a product that defend the roots of the plants and the hairs of wheat from pathogen fungi, increase the harvest because the price of the wheat in Italy is very low, and the farmer needs to improve the harvest, improve the quality of harvest because the price depends on the level of the protein inside the grains, and through the better adaptation of the plant to the soil condition. And all farms follow the quality contract like Barilla. Barilla is a very famous factory for pasta, and it propose the quality contract, and is a best way for improve the earn of a farmer. We had introduced COVERON, you can see, on the national seed dressing products. This table is right from national seed company, Italian seed company. And COVERON is a new class of products, how biostimulants products. The biostimulants products improve not only the defense, but help the plants to the growth. And in this table, COVERON is described in this way: improve the natural resistance of the plants against bad soil condition. All farmers can read COVERON mix on catalog of seed company, Italian seed company, and can ask us to have the seed directly treated with COVERON. The first experience is COVERON Trichoderma on wheat with subsiding system. Subsiding system is very important in the future because it's the best way to reduce the cost cultivation of wheat, and for receive the economic help from Europe. But this system has many risks. The first is during the sowing, the seed the seeds don't go directly on surface of the soil. And the second, the soil, the seeds are put on the dirty residues. And in dirty residues, we can have the presence of pathogen fungi. And we need to improve the power germination of seeds, to improve the resistance of the young plant in this bad condition. And this is a comparison between the seed treated with COVERON and Trichoderma with sod seeding. You can see that COVERON improved the rooting of the growth of the roots of the young plants and improve the plants the seed germination. You can see in these pictures. And the results are plants with more roots, more grow roots than compared to the chemical products, traditional products. In other phase of the growth, the effects are these. You can see on the left the plants treated with COVERON on the seeds, and COVERON stimulates more growth of the roots compared to the chemical products, and this is the effect of seed untreated with many pathogenic disease. This is a picture of this plant treated with COVERON. You can see the growth of the roots compared to the chemical products. The roots grow below the residues, and in this way the plants can grow normally, and we can obtain a very good harvest. In this case, is not possible because the plants don't have enough roots on the soil, and the plants can't absorb mineral elements like nitrogen or like phosphorus, and these plants don't have enough nutrition to have a good hairs and for obtain a good harvest. This is the results of these trials, and this is the parameters. It is the average of 4 years. Sowing date between October and November, harvest date at the end of June, the crops before wheat, soybeans or corn, and this is the harvest. You can see how can increase, how COVERON Trichoderma can increase the harvest compared to the chemical products. COVERON Trichoderma in sod seeding defends and improve the nutrition and improve the harvest of the crops. This is another experience with DURUM, wet DURUM, Triticum DURUM. The variety is a national variety, is Cesare. The results are average of 2 years, and we have a comparison between COVERON Trichoderma at dose of 20 ml to each 100 kilos of seeds, compared to chemical products, very famous chemical products in use in Italy. In traditional sowing, the effects are these. COVERON improve the mineral nutrition. Improve the mineral nutrition, mostly the nitrogen nutrition. And because the plants treated with COVERON trichoderma have always leaves largest, greenest and thickest leaf compared to the other plants. You can see the big difference between the COVERON treatments compared to the chemical products. The chemical products don't help the plant nutrition. COVERON help the nutrition. Not only nitrogen nutrition, but you can see the phosphorus nutrition of the plants. You can see the different level of growth of the roots in the plants. And this is an important question because at the final phase of the cycle, usually there is a drought condition and high temperature on the wheat. And the wheat that have more growth have more resistance to the bad environmental condition, like drought condition, and improve the stay green of the wheat. Stay green, it means more time to for a good ripening of the grains, and it means higher level of protein inside the grains. More protein, more harvest, more money for the farmer. And these are the results of this trial. You can see the different level of harvest between COVERON and chemical products, and how COVERON can improve the quality of the harvest. This is the level of proteins. The minimum level is 20 inside the grains, but 16% is very, very high level, and it means more money for the farmer. In conclusion, on the weight, COVERON trichoderma ensure good results on traditional sowing. COVERON trichoderma ensure very good results on sod seeding, and is very important in the future because the farmer are deciding to start the organic cultivation in their farm, because with the organic cultivation they have more money. The price is high than the traditional agriculture. Now I start to explain the experience on rice with COVERON against the pathogen fungi. We have a big problem in northern Italy with Fusarium fusicuroi that limits the harvest of the rice. Is a very several problem. And the results are the average of 2 years, 2080 and 2090 years. And we used COVERON trichoderma added with a special glue for rice seeds. The traditional system is the sowing of the rice in water, and this special glue allow to the spores of trichoderma, of COVERON, avoid the losses in water of the spores. We use this dosage, 150% gram for 100 kilos of seeds, plus 300 ml of this special glue. And the rice farmer put in water 5 or 6 days before sowing the seeds on water because there is the pre-germination of the seeds, and after they proceed with the directly sowing on water. COVERON rice in powder with this special glue avoid the losses of spores, and we obtain very, very good results on rice. Okay, this is the effects. COVERON rice allow a uniform growth of the plants and stimulate a good, a better seed germination compared to fludeoxonil, the chemical in use to the seed treatments on rice. You can see the different level and different uniformity of the growth of the young plants of rice. And this is the results in our experimental plot. You can see the variety is a Fedra, is a traditional Italian variety. The cases were test and treated, fludeoxonil, and the COVERON rice. We followed the advice of our national rice association, Interisi, and these are the results. You can see COVERON defends in all phases the plants and blocked the Fusarium fusicuroi, not only at in the early phases of the growth, but also at the end, when the temperature is high, very high. The final consideration, conclusion are these. COVERON rhizome maintains good control of the disease even in the late stages of development, with disease values always lower than the reference chemical product. COVERON rice has intense competition with the Fusarium fusicuroi. The common name is bacane fungus, and blocks the manifestations on rice. It is very important. We have a large market on rice for these reasons in Italy. And now I start, I show an experience on fluted corn and in presence of low temperatures. Experimental trial of the last year, 2019, and just after one month and a half after sowing, the land, the field was this. It's not rice, but it's the corn. No, it rained a lot, and the mice was all flowed with a very big damage. But amazing, after rained, the results, the effects of our treatments with COVERON were this. In our experimental plot, compared to the chemical products, the difference were very big. And it means that the COVERON help, you can see, the resistance, the natural resistance of the root of the plants against asphyxiant soil condition for excess of water. Okay, Mycorrhiza, Trichoderma help the roots to have a good nutrition in presence of a very bad soil condition. And this big difference remain in all phases of the cycle, you can see. And in these phases, we discover another features. another feature. COVERON improve the root growth. And you can see and we have many problems from with Diabrotica is an insect, and the larvae in full summer eat the root of the plants, and this larvae, Diabrotica larvae, killed the plants, the corn plants. And we are trying, I'm looking to see in which way COVERON can improve the resistance of the plants against the damage of the Diabrotica larvae, because COVERON help the plants to have a more root growth. This is the whole of Diabrotica larvae. This is our trials on corn, and you can see how COVERON always improve the root growth with good results. Okay, always good results. Okay, the conclusion, my conclusion on corn, COVERON on corn. COVERON has an anti-stress action in all phases of growth. COVERON stimulates rooting and help the plants against diabrotic damage. It's very, very important. And ensure always a good harvest. I would like to try the COVERON on the leaves in combination with a phytosanitary treatment against borers to stop the mycotoxin by the fungi that develop in injury caused by the larvae. Maybe a future way to use an alternative use on corn of COVERON. And now I show you the experience on soybean. In North Italy, there is many hectares of soybean, and it's very important because is a good soybean is a good herb for the farmer. We use a special formulation. There is only one on the market, I know, COVERON soya, because there is COVERON Trichoderma, Mycorrhiza bacteria with Azotobacter bacteria. And we made different trials with this comparison test, nitrogen fixer only, traditional products, and compared to COVERON soybean at a dosage of 130 grams each 3 doses of soybean for each hectare. COVERON soya improves the number of nodules on the roots. In Italian, the soil are very poor in organic material, in humic acids, and the soil normally have low biological fertility. There aren't many microorganisms inside the soil. We don't have enough microorganisms on rhizosphere close to the roots. And the effects are this. When we can put microorganisms, we have a very good effects compared to the chemical, the traditional system. Only Azotobacter is not enough for ensure a good number of nodules on the roots. And you can show the different level of the growth because the soil are very poor in organic material, and the effects of COVERON is very evident. And this is our picture for our different experimental plot. We have many trials, and in average, COVERON leguminose improve the number of the average number of pods for plants in all conditions. Like in this case, in sandy soil, very poor of nutrition, we have more results of COVERON soybean. This is another trial in soil with drought condition, and is very sensible. The soybean is very sensible at the drought condition. This is the different effects compared to the traditional inoculation products. COVERON improve the number of nodules and improve the growth of the roots. You can see just before the harvest the results. Okay, in average, COVERON soybean improved the good development of the number of nodules, and improve the health of the plants, because it's very common the damage by pathogen fungi like Phytophthora megasperma. And in all cases, in all trials, give a good anti-stress action against drought condition. Okay, this is our conclusion. COVERON is a good product and has many more functions than traditional chemical seed dressing products: defense, nutrition, anti-stress, throughout the plant's development cycle. This is not only COVERON doesn't have only a function, but a biological seed dressing product has many is a multifunction products, is the best way for our future agriculture. With only products we have many functions. See, if we use the chemical products, we have only a function, as is not efficient, economic efficient for the modern, for the future agriculture. COVERON is organic, is suitable for organic farming, and encourage the farmer to choose the organic farming. More money is a good chance for the future agriculture. And I would also like to use COVERON on straws to favor the transformation of crop residues. I hope that Atens study COVERON that is suitable for the treatment with liquid formulation, and for the direct treatments on the straw just before sowing, for improve the transformation, because the farmer need to receive the mineral elements, natural contents inside the organic residues. From this year, I remember, in Europe, there will be the European regulation is NEC, national emission chilling, and this regulation make a limit for the use of chemical fertilizer, and this is a problem for the farmer. The farmer will can use the chemical fertilizer only if if work for a good transformation of residues just before sowing. After the farmer can use the chemical fertilizer, and the seed, the biological, the microbiological seed treatments will be a very, very good way for the agriculture in the future. Thank you. I finish.
Thank you very much, Roberto. It was an amazing presentation, and it's always really interesting to hear about your experience using mycorrhiza and Trichoderma products, especially COVERON. We have a few questions from the audience. I will share with you. First, Roberto, this one is for you. It's about, what do you think about the application of COVERON? What are the issues that you may find with the farmer? I don't know, maybe you can share your experience.
I don't understand which is the best application of the COVERON.
No, what do you think about the application? It's easy for you, it's easy for the farmer. What can you say about that?
I don't understand. I'm sorry.
We have some people asking if what's your experience with the application of COVERON?
Yes.
Terms of difficulty.
Yeah. Yes, they are difficult for the application. Yes.
Yeah.
Yes. In Italy, we use a different formulation of COVERON. There is an industrial formulation, and we propose them at the seed factory. No. And in this case, the farmer can ask directly to the seed company the seeds directly treated, ready to directly treated with a COVERON. Okay. In other case, we propose directly our team a powder formulation for the inoculation directly on the seed just before seeds. But in the future, we give more important to the industry of the seeds to propose in a large way the COVERON.
Okay. Thank you, Roberto. Another question for you. Do you have any experience against competition products? We are not talking about chemical. Have you tried COVERON against another microbial inoculant?
Yes. We have a large experience in compared to the chemical products. Okay. And in the future, we studied the different compatibility, for example, between our COVERON with chemical products. Okay. And COVERON may be compatibility with chemical products. There is only one chemical product that killed the COVERON, is prochlorates, because prochlorates is a chemical molecules that killed all microorganisms on the soil. With the other chemical products, COVERON is compatible. We can mix together.
Great. Gorka, one question for you. They have some question about the origin of the mycorrhiza strain that we have. What's the environment in which this one was isolated? Maybe you can explain a bit more.
Yeah, the PG72, it was isolated by the IRTA, and I think it was isolated from citrus field. Yeah, there is a paper. If that person is interested on the origin of the mycorrhiza, I can provide him or her the original paper of the BAG72 discovery. That's no problem.
Great. This one is for both of you. Maybe you can share your experience using COVERON under salinity stress. What's your experience in saline soils?
For me?
Yeah. Roberto, you want to? It's for both. Do you want to
start?
start?
Okay, Gorka.
Yeah, I don't have a particular experience with saline soils using COVERON, but I have experience in saline soils using the mycorrhiza. I mean, it is the same mycorrhiza which is in COVERON, that the same mycorrhiza that we produce for on the powder format for fertigation, and it has been proved that it enhanced the tolerance to salinity. The mycorrhizal plants have a different osmotic adjustment that allows them to grow better. And in the case of the BAG72, we have seen that that's true, and it can act also as a biofilter in the roots for the sodium ion. Yeah, we have a paper in artichoke with a very high saline soil, and we improve the yield of the artichoke.
Thank you, Gorka. One last question, because we are almost out of time. Gorka, for you, can you elaborate a bit more about the adhesive used in COVERON and Covenant?
Well, it's in the patent that you can check. Let me check for the patent number. Yes, if people is interested in this patent, the better they can do is to check the numbers of the patent application. You can check either the Spanish patent or the international one, which is the image here. And you can have all the available information about how we produce that patent and the information that we can provide.
Okay, thank you very much, Gorka. Thank you, Roberto, again for taking your time and share your experience with us. Thank you to all the people who was joining us today in the webinar. You can check this session again in the future in the LiveConnect platform or in YouTube. And see you next week. We will have another session about horticultural crops. I think we are covering tomato, artichoke, and pepper, Gorka, right?
Yeah, we cover these three cultivars, and I hope to see all the people here.
Okay, thank you very much, and have a good week. Bye, Roberto. Bye.